The overall goal of this procedure is to utilize the three-dimensional culture system as a means to distinguish the benign breast phenotype from the malignant breast phenotype. This is accomplished by first plating the control and experimental cells in triplicates on a layer of growth factor-reduced matrigel in an 8-well chamber slide. The second step is to give fresh complete media changes with 2% GFR matrigel every 4th day, allowing the Acinar-like colonies to grow for 10 to 15 days. Next, the phase contrast images of the Acinar-like colonies grown in 3D are acquired at a 5x resolution. The final step is to process the Acinar-like cultures for immunocytochemical analysis with relevant molecular markers, followed by the acquisition of confocal images of in situ immunostained Acinar-like colonies at midsection. Ultimately, the phase contrast and confocal images of the cells grown in 3D culture are used to identify cellular phenotypic and molecular changes and thus differentiate the benign breast cells from the malignant cells.
The main advantage of this technique over existing methods like two-dimensional cultures, invasion or migration assays is that it can recapitulate the tissue architecture of the benign breast and its loss of organization during malignant transformation. Thus, this technique can be effectively used to differentiate the benign breast cells from the malignant breast cells.
This method can help answer key questions in the breast cancer field, such as the effect of signaling pathways on cell phenotype and in identifying new oncogenic and/or tumor suppressor pathways.
To begin the experiment,